We have a lot of speakers today focused on methyl donor metabolism, and so certainly on the front end, I'm going to hopefully provide sort of a general overview, sort of sets the stage for the overall theme of today's symposium. From my perspective, I want to talk about fatty liver disease. This is a recap, talk about disease mechanisms, what causes it from an applied but also a mechanistic perspective. Review methyl donor metabolism. I'm particularly interested in choline metabolism, review some key fundamental pathways, and then the majority of the time, however, spend time talking about three active areas of research that my lab's currently investigating. And I call these contemporary insights because I guarantee that for most of you, this is probably going to be new information. And especially for number 2 and number 3, this is the first time I'm talking about it. But we're going to review fatty acid feeding and see how fatty acid feeding with methyl donors might be something to think about simultaneously to optimize the ability of methyl donors to improve transition cow health. We'll spend time looking at some new data that we've been generating, looking at lower gut choline degradation and the formation of trimethylamine and what that might mean in terms of cow metabolism, cow health, but also choline bioavailability. And then finish with looking at lysophosphatidylcholines and how they might behave as immunomodulators. And we have some new data in neutrophils that's really promising that this lipid class has a very potent ability to influence innate immunity. In general, when we look at fatty liver disease, there's a left-hand side. This is what we're very familiar with. Overfeeding, high energy, dry cow diets, extended dry periods, high calving body condition. All of these sort of work together to increase the risk for postpartum metabolic diseases, including fatty liver, ketosis, etc. Inadequate postpartum energy intake, not just inadequate, but maybe inconsistent energy intake during the postpartum is certainly a contributor. Increased parity, second, third, and so on. Environmental stressors, we're starting to study heat stress. Anything that's going to influence energy intake certainly could be related to the prevalence and severity of postpartum diseases. Management stressors too, like overstocking, for example. More specifically, however, when we look at it from a mechanistic perspective, there's certainly been decades worth of research focused on identifying what causes fatty liver disease, and there's some nice data from Israel and the US as well showing that insulin resistance is going to be a potential contributor to enhanced body fat breakdown. Okay? Enhanced body fat breakdown increases hepatic fatty acid uptake. Right? There's inadequate fatty acid oxidation. That liver can't completely oxidize those fatty acids that are entering. There's inadequate, very low density lipoprotein secretion. Some of this is hypothesis driven, but the cow itself, relative to non-ruminants, does a very poor job of exporting triglyceride LDLs. And so you put all of this together, you get triglyceride accumulation, and certainly the severity of that sort of categorizes what the severity of the fatty liver disease. More recently, though, if you look at work that was coming from Dr. Bradford's lab and others, inflammation is a key component to this. And in non-ruminants, they've done an excellent job sort of characterizing fatty liver as being sort of non-inflammatory or inflammatory, and they actually have different names for it. In the cow, we just call it fatty liver. I have a feeling in the next decade, when we start to learn a little bit more about transition cow health, it's probably quite likely that there are also different subtypes in the cow, and certainly inflammation is probably playing a role in helping to define those subtypes. I think what's on the fringe here, this whole leaky gut conversation, the effects of endotoxin on metabolism in the liver, that's certainly something that's being talked about. And as we learn more about the gastrointestinal microbiome and the metabolites that are being produced, I have a feeling we're going to learn a little bit there as well in terms of how those metabolites are modulating liver health and metabolism. Fatty liver develops with other. This is a review back in 2004 that everybody likes to cite, but as you would expect, there's more ketosis. You get other things like DAs and mastitis, a lot of inflammatory conditions. And you get impaired fertility as well. Increased days open, lower pregnancy rates, increased services per cow. And so there's certainly an applied perspective here that this is all I'm talking about, but perhaps Dr. Overton's going to expand on this a bit more on the performance side. But today we're going to talk about methyl donors, predominantly choline, but what are methyl donors? First of all, that's a methyl group right there, CH3. I've asked that question to a couple other audiences recently, and it's important to understand what a methyl group is. And these methyl groups are important for DNA methylation, protein synthesis, right, maintaining redox balance. That's because of the activation of the transsulfuration pathway. And phosphatidylcholine is a type of lipid that can be synthesized with these methyl donors. Okay, it's a very important membrane lipid. Right, one of the most abundant. These are different types of sources of methyl groups. Not just choline, not just methionine. If you look at like the literature, I probably should have moved folate here in the order, but betaine as well. These are the metabolites, part of the methyl donor conversation. Okay, they all support the synthesis of S-adenosylmethionine, which is a universal methyl donor. I'll show you what that looks like in a second. But also, they all help support phosphatidylcholine production. Phosphatidylcholine, from the perspective of today's talk, it's a complex lipid. It's required to help form that monolayer. So a very low density lipoprotein that's assembled, made in the liver, that's exported. It's packed full of inert lipids in the interior, like triglycerides. But on the outside, you got polar lipids, like phospholipids or single lipids. All right? And the key phospholipid that's on that outside is phosphatidylcholine. If you do not have enough phosphatidylcholine, then the secretion of triglyceride within VLDL could potentially be compromised. And so dietary approaches targeting phosphatidylcholine are warranted for that reason. Finally, at the bottom, transition catalyst methyl donor status is low. I spent the last couple weeks looking up some of Dr. Erdman's previous work in the 80s and 90s, and he did some really cool work where he shows that choline rumen degradation is quite extensive, and it's not just in the free form as choline chloride, but in lysophosphatidylcholine or choline within different types of feed ingredients is very extensive. Right? He also had studies where he was post-ruminally infusing like 300 grams per day of choline chloride, and he had like 1% going to the duodenum. Right? That's nothing. Right? And so it's a severe problem that the dietary supply of methyl donors could potentially be limiting. If you look at changes in choline status throughout lactation, total choline status, phosphatidylcholine, other free choline is very low. It's lowest in early lactation relative to mid or late. And so we could potentially have a scenario where we have inadequate phosphatidylcholine production in the liver, being a risk factor for the disease. In terms of metabolism of methyl donors, I don't want to keep this fairly broad. There's a folate cycle, right? That's going to be talked about here in a second. Right, we've got a methionine cycle in the center. This involves the regeneration of methionine from homocysteine. There's our S-adenosylmethionine, our universal methyl donor, that's going to help be utilized, not just methylate lipids, but DNA, for example. We have this transsulfuration pathway at the bottom. I'm not going to get into methionine metabolism too much, but there's some new data suggesting that methionine feeding can activate this pathway here. This is important because it generates reduced glutathione. Right, and that's very powerful to reduce oxidative stress. For today's presentation, we're focused on this little metabolite here. It's a complex lipid. It's phosphatidylcholine, shown here on the left. There's our phosphocholine at the bottom, sort of in the blue. But it has two fatty acids. Right, those fatty acids, I think it's very important, and we're moving in a direction where we can't overgeneralize phosphatidylcholine. There are different types of phosphatidylcholines, depending on the types of fatty acids. Right? And they're made through two different pathways. The first pathway, at least in non-ruminants, has been established as the key driver in phosphatidylcholine production. In rodents, they estimate around 70% in the liver of all phosphatidylcholines is made through this way. This pathway does not use betaine, does not use methionine. It can use only choline and diacylglycerol as the key substrates in making it. Okay, that's very important. We don't know in the cow in terms of what proportion yet. I have a feeling that's going to be figured out here shortly, but it'll be interesting to see. The second pathway is called the PEMT pathway. This stands for phosphatidylethanolamine methyltransferase. And this pathway relies on methylation. Right? It requires S-adenosylmethionine, actually 3 of them, right, to make a single phosphatidylcholine. it can certainly come from methionine, but we also have choline oxidation that could help feed that cycle. Right? It's certainly possible. What's interesting about this pathway is it's referred to as the compensatory pathway in the liver. It's utilizing phosphatidylcholine. We can't forget that this has fatty acids from diacylglycerol as well. They've done work in non-ruminants. They show that estrogen is a strong inducer of PEMT activation, so the activation of that enzyme. And they suggested that as non-ruminants, as humans and rodents, have their offspring, they want to ensure that there's an adequate supply of phosphatidylcholine in milk and other types of choline-containing metabolites. And then finally, they have, like in humans, there's this condition that has a single nucleotide polymorphism in this gene, and those species, those rodents, are at rodents and humans. I believe this is in rodents, but I'm thinking about humans. This single nucleotide polymorphism increases the risk for fatty liver. I started to do some sort of historical research looking at sort of where this all began, and it actually started before Charles Bess and Frederick Banting, but this is a really interesting story. These were two investigators, and they were attributed to the discovery of insulin. All right, and so they sort of had a little side project, though, and that side project still involved their beagles here that had their pancreas removed. And these animals would develop severe fatty liver disease, and I sort of highlight that here, about 22% liver fat. And I don't understand the context for why they decided to do this, but early on, they were feeding them lecithin. Right? Lecithin is a phospholipid containing phosphatidylcholine. That if you stop feeding that lecithin, they developed the fatty liver disease. But if you gave them the lecithin, they only had like liver fat percent content around 3%. So one of the initial studies, at least there was a connection between a choline-related metabolite and the prevention of fatty liver disease. Subsequently, work in the early 90s really did a nice job using different rodent models where they had choline deficiency versus choline sufficient sort of conditions. And they were characterizing the VLDL that was getting secreted from hepatocytes. And they showed that in the choline deficient model, there was less VLDL. In plasma, there were less apolipoproteins that you would find in VLDL with the choline deficiency. And that these animals would develop severe fatty liver disease. So there's the triglyceride being 24 versus 4. All right? A much higher liver triglyceride deposition in those animals that were choline deficient. They also had lower levels of phosphatidylcholine. Right? And this was repeated since then, dozens of experiments. I know I could probably name at least a dozen experiments that had low levels of phosphatidylcholine and high triglycerides. In the dairy cow, there's been a large body of research that's shown that rumen-protected choline feeding can lower liver triglycerides during the transition period. So here's just a subset of those studies, right? 0 to 12 grams per day of choline, 0 to 25 or 50 grams per day of choline, or 0 versus 15 of choline chloride during the transition period. Right? Decreases liver triglyceride. Right? Dr. Overton will get into the production stuff later today, but I do want to mention that this is not always observed. All right? Perhaps when we're having this barbecue later, you can ask me a question about why that's the case. All right? Because I have some theories about why you might not always see a lowering of liver triglycerides with choline feeding. That said, there was some classic work that, soon to be classic work, right? Soon to be classic. This is 2018 from University of Florida, collaborated with Dr. Staples. And in that particular study, we were able to show Marcos was able to show they had two different conditions, sort of an ad libitum state or feed restriction, and with increasing supplemental choline ion during the prepartum. And they were able to see this decrease in liver triglycerides. We were interested in the sample set. We acquired the liver tissue, the plasma, and we have a method in our lab where we can isolate different lipoprotein fractions. Right, so that we have a fraction that contains the VLDL, and we have a fraction that contains LDL. We have others as well, including HDL. The key point here was to characterize, in that particular study that observed a reduction in liver triglycerides, how much triglyceride was in these VLDL fraction, and there was actually an increase. And so here's rumen-protected choline feeding, 0 to 25 grams per day. We have an increase in triglycerides in that VLDL fraction. In the LDL, we see the same thing. There's more triglyceride. We also had more phospholipids as well in the LDL fraction. So with our approach, we can take these fractions and then we apply untargeted or targeted lipidomics to characterize the changes in phosphatidylcholines. And we can do that with that fraction that contained the VLDL, and this is just feeding of the actual product, 0 to 120 grams per day. There was an increase in phosphatidylcholines, all right? And this is just a heat map showing that, red being higher levels of phosphatidylcholines in that VLDL fraction. In the liver, for the most part, we see the same thing, but there were some species differences where some would actually decrease. You know, you put this together with other studies, this would suggest that if you had more phosphatidylcholine being produced, that you can help potentially support the assembly of VLDLs and enhance the secretion of VLDL, enhance secretion of triglyceride. Right? And this was some work from Dr. White, University of Wisconsin. She had these neonatal bovine hepatocytes. She cultured them with choline chloride or methionine. And with the choline treatment, there was an increase in VLDL being released into the media. Right? They did not see this with methionine, however. Right? This paper goes in to show, they showed how choline and methionine also modulate the expression of genes involved in methylation. All right, so the first sort of contemporary topic I wanted to talk about is the role of fatty acids and why we should consider fatty acids within the context of this narrative. And you know, we're back to that pathway again. And, you know, there's some nice data in non-ruminants that really characterize how that composition of that phospholipid changes depending on which pathway is activated. So to recap, we've got two major pathways. We have the CDP choline pathway, and we have this PEMT pathway that relies on methyl donors. All right, they were able to use deuterium-labeled substrates, all right, able to track where that carbon was going. And then they could look at that carbon in that phosphatidylcholine pool and say, well, that was coming from the PEMT pathway, or that's coming from this pathway. And then they could look at the fatty acid composition. You know, in brief, they showed that that phosphatidylcholines that are made through this pathway, they utilize diacylglycerol. They prefer diacylglycerol that's very saturated. You'll see a lot of palmitic. You'll see some unsaturation, but it's not going to be over 2 double bonds. It's going to be oleic or linoleic. But if you use diacylglycerol coming through this pathway, through the PEMT pathway, that pathway prefers very long chain or long chain, highly unsaturated fatty acids. All right? There's clearly a difference in the fatty acid composition depending on which pathway is being used. So in my mind, I'm thinking about, well, we're feeding these methyl donors, but we also know that in a transition dairy cow, there's an increase in saturated fatty acids, oleic and linoleic, that are coming from body fat mobilization. Right? And this is sort of reducing the proportion of very long chain PUFA. All right? And this is just showing, the green showing that it's increasing in transition, these fatty acids, but these ones are decreasing. Yet these are the fatty acids within phospholipids that we should be trying to target, because these methyl donors, if you believe the non-ruminant data, and the same applies in the cow, granted it requires confirmation, the methyl donors would be increasing phospholipids highly enriched in these fatty acids. So one of the things I'm trying to think about is that if you have a scenario where you're feeding the methyl donor, but this is inadequate, then you're not feeding the entire pathway. You're only thinking about one of the key substrate. That's the methyl donor. So what we're trying to do is we have studies in the works to try to think about that, look at the synergy. And really, to demonstrate this, the need to do this research, we've done studies where we've classified cows based on their fatty liver content. So here, these cows had moderate fatty liver disease versus those that were very healthy. And then we looked at hundreds of different types of lipids. The top feature were low levels of phosphatidylcholines, and those phosphatidylcholines were highly unsaturated. And so this was sort of in support of the working hypothesis that we want to be able to target phosphatidylcholines that are highly unsaturated. You know, they've done studies in rodents where you take, this is women that have that single nucleotide polymorphism, they're predisposed to fatty liver disease, their PEMT activation is low, they have low levels of DHA containing, phosphatidylcholine containing DHA, and this is showing that as you increase the incorporation of DHA within phosphatidylcholine, this is related to activation of methylation. So So they're working in unison. So the more you can promote methylation, the more you can increase the enrichment of DHA within phosphatidylcholine. We started to look at this in dairy cows where we had different scenarios where we were abamacicline infusing emulsions with saturated palmitic acid or 22-carbon fatty acids, including DHA. And in this first figure, we had some 5x5 Latin square abamacicline infused palmitic acid being saturated, and we just characterized the changes in phosphatidylcholine when we did that, in the absence or presence of choline. Right? And so both animals were given palmitic, and these animals were just given choline. And we observed a fairly consistent response that when you gave the choline, that tended to increase a lot of the phosphatidylcholines that were highly saturated, coming from that CDP choline pathway. But we saw actually a decrease in those that were highly unsaturated that might be coming from PEMT. Same token, we took some fish oil, abamacically infused that, about 50% DHA, fairly pure source of it, pure as I could find, for the amount that we were infusing. And a 6-day infusion, and this was that phosphatidylcholine that was the #1 feature for fatty liver disease. It was most suppressed in transition cows. And with that fish oil infusion, we're able to increase the production of that phosphatidylcholine, potentially through the PEMT pathway. Not observed for saturated fatty acids, obviously, because that's, something that in the back of my mind, they've done studies where you sort of co-supplement with DHA and choline. So in this particular study, they had the co-supplementation here, and this is the production of phosphatidylcholine through the PEMT pathway. And you had the most production. But this was not observed if you just gave DHA or choline by itself. Right? And so it's about the potential synergy that we need to think about. All right, that was contemporary topic 1. Contemporary topic 2 is looking at lower gut choline degradation and the formation of trimethylamine oxide. All right, there's choline on the left, and there's trimethylamine. That's trimethylamine oxide. Okay, very similar sort of in structure. You'll go back into the literature. This was talked about in the 70s, 80s, before that. They've done studies in ruminants where you just put in choline chloride in these rumen fluid cultures, they had increased production of trimethylamine, that the rumen was converting choline to trimethylamine. Right? They've done studies that if you took starved ruminants, starved cows, and they looked at how much of that methionine, methylamine, trimethylamine that was getting converted to methane, it was substantial in a starved animal. In this particular study, they took a fed animal, and the trimethylamine that was formed would not be used for methane production. It would actually get absorbed by the animal. And in C, they were looking at some pure cultures, bacterial cultures, and looking at choline fermentation. And they showed that as choline was being degraded, and see that negative, we've got more trimethylamine. All right? So why should we talk about trimethylamine oxide? In the literature right now, it's a pretty much a hot topic in the non-ruminant world. But not just choline, carnitine can do this as well. The gut microbiome can convert choline and carnitine to trimethylamine. Right? That trimethylamine enters the animal, goes to the liver, and can convert it to trimethylamine oxide with the help of flavin monooxygenase. My grad student just informed me a while back, too, that it could also be converted back to TMA. Initially, this is a fairly inert molecule, this trimethylamine oxide. It'll go into blood, and most of it's getting excreted in urine. Okay? My grad student likes to talk about this shark. If you talk to him about barbecue shark, he'll talk to you for about an hour about the Greenland shark. And this has one of the longest longevities on the planet, but it also has one of the highest concentrations of TMAO. Well, in the past, since the 70s and 80s, when we were thinking about trimethylamine in ruminants, we haven't really talked about it a lot since then. The conversation about TMAO has sort of exploded, and that's the PubMed literature. There was initially a large body of relationships between TMAO and fatty liver disease, inflammation, insulin resistance, obesity. Higher the TMAO goes up, the lower the health goes down. All right, and so I'm thinking about it is, okay, why do we need to think about this in the cow? They've done studies, causal studies, where they've taken very unhealthy experimental models. That's very important to talk about. They're studying the effects of TMAO in animals that are predisposed to diabetes, animals that are predisposed to atherosclerosis. And in this particular study, these were animals that had a high-fat diet, predisposed to insulin resistance, If they had TMAO on top of that, their insulin resistance was more severe. This was a study where they took an atherosclerosis mouse model, and they were looking at the production of reactive oxygen species, marker of oxidative stress. The higher the TMAO, the more the oxidative stress, the more the inflammation. Again, a diseased animal. They took animals that high-fat diet animals, give them TMAO, they have more fatty liver disease. This was attributed to enhanced lipogenesis. People that have fatty liver disease have higher TMAO. And then there's one cow study. They did a metabolomic analysis. Cows with fatty liver disease had higher TMAO. Well, Billy and I were working on a project where we're abmacely infusing choline, that same trial I was talking to you about. When we abmacely infused the choline, we had more betaine and choline, but we had also higher levels of TMAO. And this was a sort of a concerning because this is post-ruminal. All right? We're post-ruminally infusing choline chloride, yet trimethylamine is going up, trimethylamine oxide. When we feed cows lecithin that contain phosphatidylcholine, we also have higher TMAO. All right? So we want to explore that. We just finished a study we presented at ADSA where we want to study the effects that TMAO has on liver metabolism and cow health. And so we have intravenously infused trimethylamine oxide. Initial data is showing that there's increased release of that TMAO in milk, and there's a substantial amount going to urine. So we're going to be able to calculate those pools and see how much is getting released. From our perspective, you know, there's certainly a potential that when you feed choline, there's this consistent positive effects. But maybe in those cases where you don't see the liver triglyceride being reduced, is it potentially due in part to TMAO? I don't know. There's also bioavailability concern, right? With post-ruminal choline delivery, right? It's possible that some of that's going to get converted to trimethylamine, right? How do we prevent that? And finally, the last topic here is focused on lysophosphatidylcholines as immunomodulators. Right? That's a lysophosphatidylcholine. There's our phosphocholine, and there's our little fatty acid. Right? I am not an immunity expert. Right? I've learned a lot about immunity in the past 6 months, but you tend to look at your data and you try to go in the right direction, and this is one of these examples. So we're learning about immunity quite extensively in the past 6 months. But to recap here, neutrophils are a white blood cell, key component of innate immunity. They help fight infection by ingesting microorganisms and releasing enzymes that help kill them. And this involves, in part, the production of reactive oxygen species by NADPH oxidase. It's called the oxidative burst. When you have that oxidative burst, these enzymes get released, help kill the bacteria. You know, this isn't, we weren't the first to think about choline in terms of immunity in dairy cows. The Bradford lab had a nice little study where he was culturing neutrophils and monocytes from cows and giving them increasing levels of choline. He was able to see an increase in expression of genes involved in choline metabolism. So more going to betaine, more going to phosphatidylcholine. He also showed that the presence of choline was able to attenuate the inflammatory response. And what he did was he had these were neutrophils looking at phagocytosis. Okay, that's the taking up the bacteria. And he showed this tendency for a linear effect and a quadratic effect with increasing choline on phagocytosis. There was a reduction in the oxidative burst with choline. And TNF-alpha, that pro-inflammatory cytokine, that was also decreasing with choline. Well, Lance Baumgarten, some of you might be able to be familiar with these data. He recently had some cows, and he wanted to mimic fatty liver disease. So he partly feed restricted them, but he also intravenously infused them with a triglyceride emulsion. Right? So their fatty acids were going up. But at the very beginning of that process, that 16-hour process, he hit them with some LPS, lipopolysaccharide, endotoxin. The rectal temperatures go up. There was no difference in NEPA attributed to the endotoxin, but you can see the fatty acids going up from the feed restriction and from the infusion of triglyceride. But that LPS did activate the acute phase response. Serum amyloid A went up. He also saw LPS binding protein went up. And so we had the opportunity to take the liver and plasma samples from this particular study perform metabolomic analyses and really use it as a discovery approach and see what's going on in terms of metabolism. Well, surprising to us, we saw this very consistent response in the animals that were given the endotoxin. And so here's our control, 0, 16 hours, 24 hours post-refeeding. And 16 hours in post-LPS, we had marked reductions in lysophosphatidylcholines with that endotoxin. Right? And this was not just one, it was all of them. Right? It was very consistent. And I'm not showing all the data, but that reduction happened by hour 4. Right? And it stayed low. Right? In transition cows, lysophosphatidylcholines are lowest in parturition. So now we have two scenarios where the potentially endotoxemia is a key feature of a transition cow. We have the induction of endotoxemia with LPS. Both of those scenarios are related to low levels of lysophosphatidylcholine. Well, why is that? Well, and this is where I tell my grad students to get in the literature and search for these things. There's a very consistent body of literature that says that people that die from a bacterial infection in the hospital, they get sepsis, they have low levels of lysophosphatidylcholine. Right? It's fairly consistent in the literature. They've done experiments where you take a mouse and you open up its abdomen and you let the gut leak in and they close the abdomen up. All of those mice die. This is survival. Okay? They're all dying from that procedure. Right? They all get sepsis. But if you gave them a lysophospholipid therapy at the very beginning, nearly all survived for some of the species. All right? On the right-hand side, this was looking at what is the mechanism of action, and they looked at E. coli counts, serial counts. And after that cecal ligation procedure, they go up. But if you give the lysophospholipids, they went down. Same thing happens if you gave them a live injection. All right? Interesting. So there's definitely a connection here. So we just recently completed this experiment, and actually these data came in this morning. We took some neutrophils, all right, neutrophils from calves. All right? Calves are certainly subject to potentially E. coli infections, right? Doesn't have to be E. coli. But we took those neutrophils and we first culture them with, we culture them two things. The first thing is PMA, this forbol myristate acetate. And the only thing you need to know about this is that it promotes the oxidative burst. It turns on the neutrophil. Right? But then we pre-cultured them with different types of lysophosphatidylcholines, and these are one of the most abundant. And we're starting to see differences depending on the type of lysophosphatidylcholine. This one, the saturated one, actually promoted the oxidative burst, whereas the 18-1 version suppressed the oxidative burst. And so this is some of the early data showing that lysophosphatidylcholines have direct effects on modulating neutrophil function. All right, so take-home messages here. Methyl donor status is low in transition cows. Choline supplementation can increase phosphatidylcholine production. That is hypothesized to support VLDL assembly and secretion of triglycerides to prevent fatty liver. To help optimize that process, we should consider fatty acid feeding. All right? How do we increase post-ruminal delivery of unsaturated fats, preferably very long-chain PUFA, omega-3s? Look at the role of TMAO on cow metabolism and health. It's hopefully something we will definitely release at ADSA this year, talk a little bit more about that. But then let's say that nothing happens with that. Let's say TMAO is relatively an inert molecule. All right? Well, we need to think about what is the extent of TMAO formation or trimethylamine formation in the lower gut, and how does that relate to choline bioavailability. Finally, emerging evidence suggests that choline and lysophosphatidylcholine are modulating immunity. All right? And there's going to be a lot more work in this area. So choline's not just about fatty liver, potentially about immune function. Thank you, my group here. Billy Myers is here today. He's been doing a lot of the choline work. He's with us. We've had some great collaborations along the way to grab some of these sample set. Specifically, I want to mention Dr. Staples. And we've had some support here. Thank you. Sure I did on time. Not bad.
All right, thank you, Dr. McFadden. That was very, very good. So, all right, I will open up. There's a couple students here with microphones, so if you just raise your hand, I will point in your general direction, and they'll walk over to you with a microphone. So, I'll open it up. Any questions from the audience?
Oh, he's always quiet.
If not, I'm going to pick on Dr. Overton. Yeah, Jose?
Can you just comment on what regulates one of those two pathways that incorporates more polyunsaturated fatty acids versus more the CDP choline or more saturated fatty acids from the non-ruminant literature? That's probably more data available.
Yeah, it's about the preference of substrate preference, all right? And so it involves this enzyme, all right, CPT, and EPT, ethanolamine or choline. And it involves that diacylglycerol is certainly going to be there. So I think it's possibly, if you look at the diacylglycerol pool, what is the composition of that pool? That could likely drive whether or not you're going to have enough phosphatidylethanolamine with diacylglycerol. We've done studies where there are lower levels of phosphatidylethanolamines that are highly unsaturated in transition cows. And so that suggests that it's not necessarily it's not the inadequacy of PMT perhaps, but the fact is that there's just less diacylglycerol that are highly unsaturated that's potentially driving that response. On the other side, the transition cow, if the same is true in the rodent, they're getting all that palmitic oleic from adipose. There's no reason to think why this pathway is not upregulated. And what's interesting is you look at some of Dr. White's work, Dr. Zhou's work, they've been able to show that choline, if you look at gene expression data, is activating this pathway. They're pretty confident that this pathway is being upregulated. But they don't always see the activation of this pathway with choline feeding. And I'm wondering if that's because just the inadequacy of highly unsaturated phosphatidylethanolamines.
Good. Any other questions?
You have 4 comments about positive effects, actually 3 comments about positive effects of choline. The one that I didn't follow is you said that choline converts to TMAO, and every time you fed more choline, that increased, correct?
So we did 2 experiments to date, post-ruminal choline infusion, increases TMAO.
But then you also made a comment that the TMAO was not very positive for the animal because it induced insulin resistance.
Yeah, right, that's a problem. That's a concern.
1 out of 3.
Yeah, well, what I'm saying is that there's consistent evidence that rumen-protected choline feeding improves health, okay? I'm not going to argue that. But with our data, post-ruminal choline infusion increases TMAO. That suggests that choline is getting degraded in the lower gut. Now, I do not know yet whether or not TMAO has these adverse effects on cow health. We're exploring that. In our study, we did a glucose challenge, we did liver biopsies. We don't have any of the data yet. And so it's certainly a question. If you ask my grad student, he hypothesizes that it's quite inert and it's not doing anything. All right?
Okay, that's the point. We don't know yet.
We don't know.
Okay, thank you.
And I also don't know what the degradation is. It could be 1% to trimethylamine. It could be more than that. If you please look at some of DeVette's work, he had a study where he abmasally infused unprotected choline chloride. He calculated 61% bioavailability. Well, that means there's the remainder, about 40%, right? That's sort of unexplained. He suggests that could have been intestinal degradation or utilization of choline, but maybe there's some TMAO going on, TMAO formation.
Dr. McFadden, along that same line, sorry, I'm behind you with a question. So that actually goes along with the question I had, which was, what do you think the base status of the other methyl groups have on each other? So sort of along that question, is that TMAO impacted potentially by the choline, methionine, folic acid, even betaine status of the animal?
So if you're thinking about whether direct effects of TMAO on the host, my guess is that because we're seeing these consistent positive effects of choline feeding, that there's likely some counteracting sort of mechanisms going on there. We also we haven't really done a good job yet sort of characterizing how much TMAO is there in a lower feeding level of rumen-protected choline. So I'm going to be fair about that. But now you may have a good question though about other methyl donors, and I make this case all the time. When we look at transition cow trials that are like rumen-protected methionine or choline trials, we tend not to think about folate status. right? We just ignore it. And it's never mentioned anywhere. And so there's certainly interactions, because when you look at those pathways, these cycles are all connected, right? So I think you're going to probably mention something about that. Good.
Other questions? We still got about 7-8 minutes. All right. Thank you very much. Let's give one last round of applause.